And I realized I have like the sleepy slot after lunch, but just before the coffee break. But bear with me, and we will do this together. And hopefully I can woo you with some cool images. So my name is Madeleine. Thank you for the kind introduction. And I'm going to talk about how we model cancer invasion plasticity in glioblastoma. So I am a third-year PhD student in Sven Nylander's lab in Uppsala. And we work primarily with glioblastoma, which is the most common and most aggressive primary malignant brain tumor in adults. So primary because it's the kind of tumor that arises straight directly in the brain. Other tumors can metastasize to the brain, but this is the most common tumor that arises in the brain. And in Sweden, about 400 people get this diagnosis every year. And from numbers I can find, roughly 20,000 people in the whole of Europe. So it's kind of rare. But and the age of diagnosis is between 55 to 75 years old. And median survival is roughly 15 months. So the prognosis is really, really bad for these patients. And here is just a graph showing that from a selection of different gliomas, it's oligodendroglioma, astrocytoma, and we see that glioblastoma has the absolute worst prognosis with a 5-year survival rate of about 7%. And it usually occurs in the frontal or the temporal lobe of the brain. And treatment options include surgery with maximal resection, radiation, chemotherapy with temozolomide, and tumor treating fields. And current therapies reduce tumor burden, the patients will eventually relapse. So this is a graph comparing patients that had radical surgery, non-radical surgery, and just a biopsy. And we see that surgery doesn't really do much for these patients in terms of survival chance. And that's because treatment is very complicated by the diffuse and infiltrative nature. So we see that these tumor cells spread all throughout the brain in a multitude of different ways. So they often spread, let me put the laser pointer on here. So they often spread through the other hemisphere, through the corpus callosum, so along matter tracts. They spread through by other anatomical features such as blood vessels, they spread to the meninges, and also diffusively straight to the brain parenchyma. Oh. Okay. Yes. So essentially, there's a number of ways that these cells can spread. Perfect. Thank you so much. And since the theme of this conference was a multi-scale view of the brain, I thought that I would offer a multi-scale view of invasion. So traditionally, when we talk about invasion, we talk about it on like a tissue level dissemination perspective. So essentially what I just talked about, that cells spread throughout different anatomical structures. And we've also shown in our lab that different patient samples from our in-house spider bank, they grow in the brain in different ways. So some tumors bulk and they have a lot of perivascular invasion. Their tumors grow very diffusively. So they actually spread throughout the whole brain, but there is no clear bulk. Then there are also more mesocale perspectives on what's going on. So this is a paper by Fasalt, which describes something which they call oncostreams. So it's part of the tumor tissue where cells along the same direction to form the streams. And then we also have more complete cell descriptions on how the cells are migrating through the brain. So this is a paper describing how cells hijack neuronal mechanisms of migration. And in an attempt to kind of tie all of these perspectives together and describe how cells disseminate through the brain, we established a system for modeling glioblastoma infiltration in real time in the mouse. So what we do is essentially we take patient-specific GFP-labeled cells and we inject them orthopically into mice, and then we let the tumor establish for about 60 days before we take out the brain and we slice the brain with the tumor in it, so an established tumor. And then filter with a fluorescent dye. And then sometimes we drug the slices and we image it. And we have data that looks like this. So we have a whole view perspective in the brain, but we can also zoom in to get single cell behaviors over time. And I should also take the moment to say that all of the experimental data and images that you will see today is generated by my colleague Hitesh. So he does the experimental work and then I try to analyze the data. Just a disclosure. And then we can look at the data just both full scale, but also in different example regions. And we can look at the heterogeneity between different regions. So we see that cells have different shapes, different sizes. They move at speed. Some of them just migrate diffusively, as we talked about. Some of them migrate along vessels. And then we compare between patients. We see, so this is the model that I showed you. And then we see that in another patient model, we have these cells that have super long protrusions. And then in yet another model, cells grow or more kind of reminiscent of collective invasion. But regardless of this heterogeneity that we saw, we managed to kind of tease out some recurring patterns of invasion over time. So we saw something that has previously been described as branching migration, and it's when cells have multiple protrusions in the direction of migration. So that looks like that. And then we saw cells, which we saw before, that has one unipolar protrusion in the direction of migration. Yeah. And then we also see cells with a more fibroblast mesenchymal phenotype. We see cells migrate parallelly. And then, so these are some of the recurrent patterns that we saw. And then in the end, we decided to, for simplicity, strategize into 5 different groups. So the ones that I described, plus brown cells, which we saw present in the slice. And then what was encouraging to see was that some of these morphologies has been described before, has also been seen in intravital microscopy. And if you're not familiar with intravital microscopy, it's when you implant a small window into the skull of the mice, essentially, so that you can observe migration patterns in vivo. So that kind of made us a bit more sure that this was just not an artifact of the slice culture, but we're actually recapitulating what's happening. And then the natural thing to do when you have this type of data is that perhaps you want to track the cells over a long scale of time. So all of the tracks that you've seen here now is just manually annotated by my colleague, but it would probably be interesting to track multiple cells and see how they behave on a population level. So next, we developed a pipeline for automatic cell tracking. So we do some very simple cell detection, nothing fancy really, but we do blob detection, image manipulation, and then some coordinate pruning to go from images to coordinates. And then to track cells over time, we essentially predict the position of a cell at the next frame, and then we match the predicted position to the closest cell in that subsequent frame. And we predict this position by a very old school algorithm called Kalman filtering. So that's a way of predicting object positions in time. And we assume constant velocity, and then we can just calculate how the position develops over time. And then at each time point, we also classify our cells into one of these 5 morphologies that we've grouped. So we trained a very small convolutional neural network to give each cell a label at each time point. And then we end up with data that looks like this. So on the z-axis, you have time, and then you have x and y coordinates. And each little string is a cell track over time labeled by its morphology. And if you're ahead of me in thought right now, you might see that cells don't have the same colored label across time, meaning that they switch mode of morphology or mode of migration and morphology over time. So this is something that we've already observed qualitatively. So here we see a cell that first is attached to a vessel, but then migrates diffusively for a bit and then attaches to a vessel again. But now that we also had quantitative tracking data of cells over time, we could describe this in a different way. So we described this with a hidden marker model to describe how the probability of transitioning between different states over time. So for each, this looks weird. Okay, but for each, essentially for each patient model, we get a unique transition matrix of how the transition between these different morphologies look. And if we take a step back, this concept of switching between morphologies isn't really foreign, because we already know that cells can switch from, for example, collective migration to single cell migration, from amoeboid to mesenchymal migration, and so on. And so we know that tumor cells adopt a variety of migration strategies depending on tissue properties and external stimuli. And so migration mode plasticity is probably a very important adaptation skill for tumor cells. Weird. But essentially, what we wanted to do is essentially not only look at how these transitions happen over time, but also ask questions such as which transitions are completely stochastic and which transitions are the effect of external stimuli. So in an attempt to do this, we tried to describe the local milieu of the cell with 3 different measures. So we tried to describe the distance to the nearest vessels, and this could be interesting because it's been previously reported that glioma cells proliferating near vascular branch points have been seen to proliferate near vascular branching points. And we also know that vessels is a source of gradients of nutrients and oxygen, essentially. And so we had already developed a UNet for segmentation of vasculature in the slice. So, and then since we label the vasculature with lectin, we don't only label fluorescently label blood vessels, but we also label microglia, which is a immune cell in the brain. And then we, several times we observed tumor cells co-localizing with these microglia, and we saw that this had a big effect on the morphology of the cell, probably some scavenging behavior. So we found a way of also automatically detecting this by training a neural network. And then we also developed a measure of polarization of tumor cells in the region. So we looked at regions that were either the cells were completely aligned, or regions where cells were moving more stochastically. And the idea was that this could be some sort of proxy for ECM structure or other local structures not currently visible, because as we heard in the last talk, it's very crowded in the brain. But what we visualize is essentially only the vasculature, the tumor cells. So there's a lot of things that we don't see. But we thought that it might serve as a proxy for that. And so what we wanted to do was essentially to couple the features of the cells that we described to transition probabilities. And it's not supposed to look like this. And so in order to do this, I'm going to quickly take how we covariate hidden marker model, the GlioTrace. So in our case, we denote the hidden variable as the latent mode of migration of a cell at time t, and outputs from the convolutional neural network as the observation evidence, essentially. And so usually a hidden marker model is defined by some kind of initial distribution across the states, a transition matrix describing the probability of transitioning, and also an emission matrix. But in our case, we only use the CNN outputs. And then this is trained with an EM algorithm called Von-Welsh. So in the expectation step, we take our track data and we loop through each cell. And we take the softmax probabilities and the features that I described. And if you've forgotten, these are how the tracks looks. And then we pass these to forward-backward algorithm to calculate the log probability of the observed sequence. So that looks like that. And then we get, for each sequence, we get the posterior probability of the hidden state at each time point, given the full track. At each time point, we also get the transition probabilities between each state. And then we pass these to the M step, and we pick the parameters of our model, and we update the transitions by fitting a multi-logistic regression to the data for each from state. And then we give the parameters back to the E step. We will calculate the log likelihood of the observed sequence, and then we iterate until convergence, essentially. So nothing fancy. Let's see. So essentially, in order to couple the local features to the transitions, for each transition from class I, we model the probability of going to class J using logistic regression. And then we can look at how the probabilities change based on whether the feature is present or not present, or high or low. And I think I'm gonna this, because it looks really, really weird. Okay. Give me 30 seconds. I'm gonna reopen my slides. Yeah, or I don't know what happened to my slide. Didn't look like this before.
This one okay?
No. Something with the formatting. Okay. Okay, so we'll just look at one example. Yes. Okay, yes. And then we can make some trivial observations and some possibly interesting observations. So, for example, so this is what we get. We compare the transition probabilities given the 90th% of the feature and the 10% of the feature. And each row here is a probability from a state to another state. And here we have also included self-transitions. So, for example, we see that when vascular distance is very high, the probability of transitioning or staying in the perivascular transitioning state becomes very low. So that's like self-explanatory. And then we also see that cells that do co-localize with microglia, they tend to switch a bit more. So that's indicated by the red bars here. And yeah, so I apologize for this. There's something weird with the formatting here. But we also see that cells in polarized regions tend to switch less. So they tend to stay in their original morphology, which was rather surprising to me because when we looked at turning angle distributions of these morphologies, we see that they are all fairly persistent over time. So each time they take a step in space, they roughly stay at the same path. So it might indicate to me that switching is somehow coupled to changing direction, for example. But that's also speculative. Yeah, but so in the end, what we developed grew into a full framework that is now also on GitHub. So if you know anyone with imaging data that needs cell tracking and morphology analysis, please send them to me. And yeah, to summarize, glioblastoma cells in the brain exhibit heterogeneous and plastic cell migration strategies. Cell migration can be stratified in five groups based on cell morphology, at least in our data. We have established a framework for cell detection, tracking, and classification in live image brain-sized tissue. And we can model invasion plasticity using a covariate hidden Markov model. And we see that features such as polarization affect transition rates in a measurable way. And with that, I want to thank everyone involved in this project. Hitesh, who did all the experimental work, and people who built the model with me, and you guys for listening. So thank you so much.
Thank you so much.
Yes, thank you.
Yes, question, whatever.
Can you say something about implications of the relative fraction of the different migrations, subgroups on patient outcome and treatment?
Yeah, so that's a very good question, and frankly, there's not a lot of measures in glioblastoma overall that can stratify patients right now into different, based on overall survival. So regardless of whether we look at molecular data or morphological data, we don't have really any meaningful measure to stratify patients like that. But generally, when you talk about migration, some cells adapt more developmental-like strategies. So for example, neuronal-like strategies for migration, and other cells are more injury-like, so like wound healing, fibroblast migration. And I would guess that cells that have a more developmental-like phenotype and also transcriptional identity are perhaps more resistant to, or less resistant to therapy, for example, than the injury-like cells. So that's kind of what we're trying to encode here also, like adaptability as a proxy for resistance, essentially. Yeah, but a great question.
Very interesting. I wonder, sorry, maybe I lost the information. When you say in slice, you mean how was the preparation?
Yeah.
And then the second, you also mentioned that this would be maybe very relevant also to apply that in vivo. Have you started that also? Or cranial windows, for example?
Yeah. So the whole idea of the slice methodology is to essentially replace the cranial window method because it's very invasive and it's for a very short time period. So with our method, we can visualize or we can image for up to 6 days. And we believe that it's a bit more ethical, albeit marginally.
But you have to. Not that you don't do anything to the animal, right?
No, of course. It's all relative. But this is how we, so we inject.
So you keep the slice alive.
Yes, exactly. Exactly. Yes. But thank you for your question.
Yeah, also super nice. I'm just curious. So they're never still, like, you don't have a state where they're not moving?
So we see that cells that are rounded up are essentially immobile.
Would this
So
give them a more attractive state? I mean, in terms of kind of want to slow them down, I guess, send people to surgery. They would to.
Yeah, exactly. Yeah. Yes.
Yeah. Thank you for your talk. Do you see any genetic differences between glioblastoma cells that are moving and the ones that are?
That's a very good question. I haven't looked into that. Right now, we're kind of after looking at the transcriptional identity in real time of different morphologies. But it would definitely be interesting. Yes.
Yeah.